talking with microbiologist Linda Rhodes. During her 45 plus years as a scientist, Linda Rhodes conducted research in the areas of aquatic toxicology and microbiology. Early in her career, she was involved in work correlating the presence of anthropogenic chemicals in the marine environment to the presence of lesions, including tumors, in wildlife populations of fish and shellfish in Puget Sound and other U.S. coastal areas. When salmon along the west coast of the U.S. were listed under the Endangered Species Act, her work refocused on infectious diseases that affect both wild and hatchery-reared fish. The advent of next-generation sequencing allowed her to apply that technology to describing microbiomes of killer whales as potential health assessment tools and to environmental microbiology for evaluating urban stream restoration and coastal marine waters that experience ocean acidification. These seemingly diverse areas have integrated her knowledge of large and tiny critters and ecosystems in the Pacific Northwest. Closer to home, Linda applied her scientific knowledge by participating in Island County's Environmental Health Assessment Team, Marine Resources Committee, and Conservation Futures Program Citizen Advisory Board. Linda has a PhD in molecular and cellular biology from the University of Washington, and she has lived full-time on Whidbey Island since 1987. She feels extremely lucky to have been a part of both the scientific community and her home community here on Whidbey Island. Thank you for being with us, Linda.
Oh, my pleasure.
I'm really excited to talk with you about microbiology because it's something that I know little to nothing about.
You're not alone. I think that's true for most people, except in disease. And that's the one thing they do know about microbiology is that many bacteria or many microorganisms that are capable of causing disease. But most of the time, people probably don't think much about it.
Yeah, which is interesting to me because it makes me wonder how you got started in microbiology. Like, how did you decide to study that in the first place?
Right. Well, actually, I was originally interested in molecular and cellular biology. When I went to college, long time ago, back in the early 70s, there wasn't a whole lot of molecular biology that was part of the curriculum. But once the advent of the, techniques and technologies to start looking inside of cells and actually looking at DNA and RNA and understand how they functioned in cells, to me, that was just absolutely fascinating. So that's kind of the small scale universe. So I was actually one step lower than bacteria and many of these other microorganisms. I was interested in what was going on inside of the cell. But in order to do those kinds of studies, you actually use quite a bit of microbiology. Those are the tools that were developed by many people who have Nobel Prizes for their work. They used viruses and bacteria as tools to study what was going on inside of cells or how DNA was being translated into RNA and how was the RNA being translated into protein. So I kind of came through the back door in microbiology, really using that as a way of looking inside of the cell. And that's actually my PhD work it was focused on gene expression. But so by the time that I got to looking at bacteria specifically as a subject of study, I was already familiar with how do you grow them, what are they doing, what are their properties, what are their characteristics, and I began to realize that particularly in the study of diseases, there's an interaction between the bacteria, and I'm going to focus on bacteria because I'm not a virologist. I mean, when you say microbes, it includes anything that's microscopic. So that includes even fungi. Some fungi are single cell, and you need a microscope to see them. There are even organisms that are called eukaryotes. Probably the old classifications, prokaryotes and eukaryotes. And there are actually eukaryotes that are single cells. Amoeba, there's a good example. So I'm really focused on bacteria. And once I began to look at disease and caused by bacteria, you begin to realize there's this interaction between that bacterium and the host, the animal or the organism that's carrying, and that the disease state is really kind of a negotiation between the host and the bacterium. So it's not just warfare. It's usually some sort of interaction. And probably the analogy is when you look at larger things that you can see walking around and the interaction of an animal and its environment and other animals. So it's the same thing that people, ecologists, study, but it's actually on a smaller scale. And once I began to learn more about how do bacteria function, how do they talk to each other, how do they talk to the host, it's a fascinating world in itself.
Yeah, so you mentioned that that is fascinating, and I like the idea that it's kind of like ecosystems on a micro scale. That
exactly.
There's all of these relationships that are happening, that if you start to look at behavior and relationship, you can kind of get a larger picture of what's going on in like this tiny community, which I definitely want to talk about, what we can learn about viewing things from the microscopic lens. But I am curious, just as a basis, how is the discipline of microbiology defined? Because you mentioned cellular biology, which is study of the cell and everything happening inside of it, right? But what is microbiology, and what are the kind of different disciplines within microbiology that people study? You said bacteria is your specialty.
Right. And viruses, that's another kind of, that's another microbe that is entirely different from bacteria, because those viruses are completely dependent on their host. They have to be inside of their host. And there are actually even viruses that infect bacteria. So, everybody feeds on everybody else or, is dependent on others. And so those viruses that infect bacteria, they're called phages, and they can actually blow up a bacterial cell. And there's been a growing movement about using phages as opposed to antibiotics, chemicals, to suppress growth of bacteria in a disease condition. Trying to use phage to do that work, because phage are very specific. So there's viruses, which include phages. There's mycology, the fungi, that are another, again, microscopic, although they can grow to be macroscopic if need be. There's actually another kingdom in the world that's called archaea. And they're kind of in between the bacteria and the eukaryotes in terms of how their genetics are organized, but they're single cell, they're microscopic, they're like bacteria. I think about bacteria in, say, ocean conditions. They've been initially were characterized under extreme like rift vents, where you have boiling high temperature water with lots of sulfur coming out from the Earth's crust. So that's kind of an extreme condition. And they tend to be identified in those extreme conditions. But what we're learning with the tools that we have today, that they're actually fairly widely distributed. Wow. So so the archaea, and they're not necessarily not so well known for causing disease conditions, but they are very much present in the ecosystems. So, and then there are some parasites, malaria parasites, and they're microscopic too. They're flagellates, and they can get inside your blood. A mosquito can pick it up and inject it into you, and when they're taking a blood meal, and they get into the bloodstream, and they are very happy to proliferate and go through their life cycle in a human being. And of course, they cause a huge amount of death, human death, and grief. Yeah. So it's a huge range of microorganisms that are out there in the world that we often don't know much about until they cause an inconvenience for humans.
Right. So would you say microbiology is the study of anything biological in nature that humans can't see with the naked eye?
Yeah, that's a very good definition.
Okay. Yeah. So it's all about us, right? And then we study it if it affects us. Is that?
Yes. Well, it's the motivation.
Yeah, totally. So I'm curious, what makes a eukaryote, and then what makes bacteria?
Well, a lot of that in the old days, that was defined by whether or not the DNA was enclosed in an envelope or had a membrane surrounding it. And so eukaryotes have their DNA is compartmentalized inside of the cell and surrounded by a membrane. And how the information that's in that DNA gets out into translated into RNA, things have to go in and out of the membrane to get into the DNA. In the case of quote, prokaryotes, their DNA, their genetic component is not enclosed in a membrane. It's kind of present, and it can be in a circle or it can be in a long loop, but it's sort of just sitting in the soup of other kinds of molecules that are inside of the cell. There's still things that control what parts of it get turned into RNA and then into protein, but the membrane is not part of it. So that was one of the key features that was used early on to differentiate between the prokaryotes and the eukaryotes. And of course, now as we know more and more, there are a lot of people saying, oh, we need to get rid of these old categories. But it's useful, for classification purposes, it's useful.
Cool. And then, so since you're an expert in bacteria specifically, what is bacteria? What's the definition for bacteria?
It's organisms. Those are organisms that have a particular kind of cell structure to the wall that encloses the entire organism. So it's a single-celled organism, and there are just a few kinds of wall structures that are identified. They can be free-living. And in fact, the vast majority of them are free living. They don't depend on a host, or they don't have to be inside of another host, but some are capable of living inside of a host. So that would differentiate them from viruses, which are much, much smaller too. But their capability of being free living. And then there are many other features that would, for instance, they don't have any kind of sexual recombination. I mean, sex is a way of mixing up genes and producing something totally new. And they don't use sexual reproduction. Fungi use that. Yeast, there's a great example. Yeast is a microorganism, and they actually do have sexual forms that recombine. For bacteria, they are capable of actually swapping pieces of DNA with other bacteria. And they can also take up what are called extra chromosomal. So their chromosomes define everything that represents that bacterium. It defines the membrane and all of the structures inside of the cell and all of the molecules that it might produce in order to stay alive. But there are also smaller pieces of DNA that are independent of that chromosome that are called plasmids. And those can actually replicate inside of the bacterium to great numbers. And a lot of times, antibiotic resistance, for instance, can be carried on a plasmid. And depending on the kinds of pressures or stresses that the bacterium receives, it could actually increase the number of plasmids to a high level that could confer antimicrobial resistance, which, of course, is a big concern. That's not something you really see so far, nobody has really observed that kind of phenomenon in archaea and some of these other parasitic microscopic parasites. So it's a little hard to say, here's exactly those features, but those are characteristics that are somewhat unique to bacteria. Cool. And I know there's always exceptions. As soon as you say, these are the general features that you look for, then there will always be an exception to the rule.
Yes.
But the size is the big thing.
Okay, size, and then it doesn't, size and
independent living,
and they don't generally use a sexual reproduction. Right. They reproduce without that, right? Or not reproduce, they replicate.
They replicate, right.
Yeah. So why, I know when we first spoke, I think you mentioned this, and I'm curious about this. Why is microbiology called the second genome? What is that?
It's not microbiology itself, but there's a term that has come into use within the last 15 years or so, maybe 20, maybe 20 years, called the microbiome.
Oh, yeah.
And so the microbiome obviously encompasses all of the microorganisms. And then a biome would be, it's kind of the community or the consortia of microbes that exist in a particular location. So for humans, there has been, the National Institutes of Health has had the Human Microbiome Project that was started around 2007 or so. And their goal was to identify all of the microbes that live on humans. Now, they focused really on bacteria, but they are obviously looking at other kinds of microbes as well. And in that case, because they focused on bacteria, the understanding was that these microbes that live on us are not just necessarily hitching a free ride or getting a free lunch off of us, but that they actually communicate with our cells and talk to our cells, and that there is an interaction between those two. And then the Human Microbiome Project started to look at different parts of the body and compare that across people. And what they discovered was that the bacteria that live on, say, your arm is more similar to somebody else's arm than it is to the bacteria that live in your nose or the bacteria that live in your eye. So your different parts of your body are kind of different habitats for bacteria.
Oh, interesting.
And across people, we have shared habitats. We have similar habitats, but as anything, anybody's ever studied biology, there's always diversity. And it turns out that you as an individual will have a microbiome that has differences from my microbiome, and therefore differentiates you from me. And you've probably seen some, television shows, where they start incorporating this into determining, okay, we can get fingerprints or we can do a DNA match. And then some have even started to talk about microbiomes as a way of saying, oh, we looked at somebody's keyboard and we can see that that person never touched this keyboard because of the microbiome. I'm not sure we're at that level of being able to use it as a tool, but because these bacteria, and the estimate is that for every one of your cells as a human, there's 10 bacterial cells. So we're outnumbered. They're tiny though. They're very tiny. Our cells are much bigger than them, but we are basically outnumbered by all of those bacteria. And because they interact with our cells and actually change how our cells are functioning, that's where the second genome concept came from, is we can look at our own genome to try to understand how it's being used to make us an individual or to make us healthy or not healthy. But we can't ignore the fact that there's a second whole sets of genomes that is interacting with us and affecting us on a daily basis.
Oh, that is super, super interesting. I think I didn't realize that I knew that we were outnumbered by bacteria, in our own personal ecosystems, but I didn't know that they were communicating with our cells and changing cell behavior necessarily. And what I think is really interesting about that is that I think on a very basic level, and you mentioned this earlier, that human beings generally don't think about bacteria unless it's interacting with us in some way that causes disease. But bacteria is actually interacting with us all day long, all of the time.
All the time.
And so I'm curious, how much of, bacterial study is, I guess maybe a lot of it, maybe the study is about disease, but, what percentage of bacteria is causing disease, and what is the rest of it doing, on me?
It's hard to come up with a percentage, but I'm going to, speculate that it's down around the single digits and maybe fewer. One of the things to keep in mind is that the early studies of bacteria didn't have the advantage of all the genetic tools we have today. So when Robert Koch back in the 1780s or whenever, kind of the grandpappy of doing a lot of bacteriology. I mean, and that's kind of, under microbiology, you can have bacteriology as just the focus on bacteria. They had to culture things. They had to grow them up. And so as a result, there are a few bacteria that are known to cause disease that actually grow very well in culture. Now, there are many others that don't grow very well in culture, but they've gotten a lot of attention, and an awful lot has been learned about what they do. And they're kind of studies of other bacteria will often reference back to those. E. coli is a great example, Escherichia coli. It's in your gut. I know everybody goes, oh my God, E. coli. But the thing is, it's normally there. It's just a very small percentage of the gut bacteria, and it does do a job. And when it causes problems, there's some other stuff that goes on when you get lettuce or something that's contaminated with E. coli, and FDA tells you, get rid of all pull the stuff off the shelves and get rid of it. But it is there normally. And as long as you don't have the problematic forms of E. coli, because there are a lot of different strains. Of bacteria. And as long as you don't have a strain that's carrying a toxin, it's not an issue for you. So the other thing that's hard, so there are a few bacteria that we know a lot about, and we know how they make a living. There are a lot of bacteria that we don't know how they make a living and what they do. And that's really going to be the limitation in our ability to use the great genetic information that's come available most recently. So, for example, you can take a sample of water, filter it out, extract all the DNA that's in there, and that represents DNA from a whole bunch of different kinds of bacteria that were in the water. Then you can use a technique called PCR, polymerase chain reaction. I think everybody knows about it these days. You can amplify. PCR allows you to, in a tube, actually amplify the amount of DNA that's in there. And now you can actually sequence all of those amplified strands and get millions and millions of sequences, which you can now compare to big databases to see which bacteria can be identified in there. Now, I do that. That's what I do with water samples. It's a great tool. And I call it building a phone book. Basically, I'm going in there and I'm putting names on what I've seen or what the sequencing has seen. But if you think about a phone book, you pick up a phone book and you go, oh, look at all these people that are in here. Some have similar names, some have different names, but you don't know anything about those people. You don't know who is the policeman, who is the nurse, who is the teacher, who does what. And so in many ways, it just having that phone book doesn't really tell you a whole lot about the functioning of that community. In order to know that community, you need to go there and say, oh, you're so-and-so and you do this. All right, I understand your role in the system. And so it's a similar thing.
In the environment in order to understand the functioning on that level also? Is that what you're saying?
Well, and you need to understand what each bacterium, well, you don't have to understand each bacterium, but for a particular bacterium, you need to know how it makes a living. So some bacteria, you have to have very simple carbohydrates. They can't take more complicated carbohydrates like mucins and the stuff you see sloughing off of kelp fronds. They can't use that. That has to be already broken down into very simple, say, sugars or very simple carbohydrate form, because they don't have the enzymes to do that degradation to do that. So, and then what do they produce as a byproduct or as a metabolite from their work or from their daily living? So that's where it really is going to improve our knowledge tremendously if we know what bacteria are doing. Now, you can infer that from the genes. So that's where genome sequencing, that's a different kind of high second generation or next generation sequencing. You can sequence the entire genome of a bacterium and you can say, oh, here's a gene that actually it looks like it produces an enzyme that can break down those complex polysaccharides. So maybe it's not a surprise that whenever you have an algal bloom that occurs, they make lots of polysaccharides. And then now if you see that bacterium present, you figure, they're taking advantage of all of these polysaccharides that those microscopic algae are manufacturing.
Right. But I guess you have to be monitoring the behavior in relationship, right? It's like of each thing you've identified in the phone book in order to understand its functioning in the larger ecosystem. But it's on a microscopic level. Like I'm thinking of Steve and Marion and how they do plant identification and how going out, and seeing just even like wildflowers and native plants and the discovery process of that. And then being like, well, what does it mean that this plant is here? What else is going on? What are the other conditions that need to exist in order for this plant to show up in this place, in this time, in this way? But you're doing it, and microbiologists are doing this with things that we cannot see, which like not normally. And so I think that's so interesting because I'm just curious how you observe.
It's through right now, a lot of, at least my work is focused on looking at the DNA that's present. So you're doing it indirectly. You're not really taking water and looking at it under the microscope.
Right.
You can do that, but you can't, that's very time consuming and you don't have all the tools available to you to do that. Whereas by using the DNA that's present, then you can start saying, there's so-and-so, and there's so-and-so, and there's so-and-so. And then I'm totally relying on the probably thousands of years of work that people who study individual bacteria in the laboratory, and they put it under different conditions. They give it different culture conditions. They combine it with other microorganisms or other bacteria to see how do they interact with each other. What I'm inferring that this bacteria might be doing is based on their work. So there's a huge reliance. I mean, I'm just stealing information from other people's work to make kind of a conclusion about what I think is going on. And there are many people who do what I do. So it all relies, it's all integrated in that sense. And that's where scientific publications are incredibly important as a resource. And also these large genetic databases that the NIH and other countries, the EU, are supporting, because we wouldn't have anything if it weren't for all that work.
Yeah, everybody sharing their work, I think, is extremely helpful. You're right, because you can use the observations and kind of create identify trends through observations from other people's research. Right. I'm curious, you're talking about identifying trends. I'm curious about your work with NOAA Fisheries around the salmon populations in the Pacific Northwest. And I think people listening would be very interested in learning what you discovered, why you were working on that, what kind of led up to you working on that, and then what you and your team discovered while working on that project. Okay.
Just in terms of the bacterial work, as I mentioned earlier, that you mentioned in the intro, once salmon became an ESA listed species, then there was tremendous interest, particularly in doing captive broodstock rearing. So that's really where you take like the redfish lake sockeye from Idaho, from the Sawtooth Mountains. That was certainly that's one of the endangered populations, and it was so low that NOAA Fisheries and determined that they needed to bring them into captivity in order to breed them and rear sufficient numbers. They couldn't just rely on them naturally migrating back up the 900 miles up the Columbia River and the Snake River to get to where their home grounds are. So when that happened, then there was concern about, okay, now we've got animals in captivity, and this is true for all captive broodstock rearing programs. Now we got animals in captivity, so that's where your potential for disease, observing it and treating it, is much higher. So that I worked on and this happens to you as a scientist, you have to really focus on one thing. So
Yeah.
I focused on one disease that was not just in the sockeye salmon, but is also a really common problem for Chinook salmon and somewhat also for coho salmon, and to some degree steelhead, which is called bacterial kidney disease. And it's caused by a bacterium, as the name says, and it continues to be a chronic problem in hatcheries, both conservation and production hatcheries. And the reason why is that as it turns out, I probably wouldn't have chosen it if I'd known this at the beginning. But this is a bacterium that is so well evolved with its host that it is very difficult to separate it from the life cycle of the host. And I'll explain what that means.
Yeah.
The bacterium, most of the time we think about transmitting bacterial pathogens through what's called horizontal transfer. You give me something because we're in the same room together or we've shared some implement. And Renibacterium salmoninarum, the bacterium that causes BKD, bacterial kidney disease, can do that very well in water. That's one of the problems with water, is it's a wonderful transmission medium for pathogens. It's much better than air. But this bacterium can also be transmitted vertically. That is, a female can, as she started, a female salmon developing eggs, we've actually used a marker that identifies Renibacterium. We've been able to detect them in the kidneys, or not the kidneys, but in the ovaries of females a year before they actually spawn.
Oh, wow.
They can be infected, and it'll be sitting there waiting in the ovaries. And when that fish spawns, the bacterium can actually get into the oocyte and be inside of the egg itself. So when she spawns fertilized eggs, the bacterium is already sitting there waiting for the embryo to develop. So that's called vertical transmission as opposed to horizontal transmission. And surveyed fish throughout, juvenile salmon that have migrated out from freshwater into saltwater. And we have seen lots of infected salmon. At that point, the infection is primarily in the kidney. That seems to be a good target, that it likes to go in there, sit in there and hang out. And it probably goes with the fish through its entire life cycle. This is the Renibacterium is able to go into a very quiescent state. It doesn't have to keep reproducing. It just sits there and bides its time. And there may be some interaction between the fish and the bacterium that keeps the bacterium from proliferating. Because what it doesn't want to do is kill the fish, right? It wants to live with the fish, go out to sea, come back. And at that point, at least for the females, wants to be passed on to the next generation of fish. And the times that it causes mortality and clinical signs is when you have fish in a tank or in a raceway and you have this very high density situation. Something might not be quite right. Chinook are hard fish to rear. They are very fussy. And unfortunately, we have a lot of Chinook hatcheries. But that's when it becomes a problem. There's some kind of a stressor. The fish doesn't have its full immunocompetence. And then the bacterium responds to that by growing and proliferating. And we don't know, you don't want to be teleal, you don't want to assign an intent to the bacterium, but there's probably some signal to the bacterium that this fish is going downhill. So I better try to just replicate as much as possible to make sure I'll get distributed horizontally, right? As opposed to
right. Because horizontal distribution is a possibility for this bacterium. It's just but it also can vertically transfer. So it has right. Okay. And so in this bacterium and this disease occurs naturally in the wild.
Yes. It's endemic. It's widely distributed among salmon populations.
Mm-hmm. But in hatchery situations where there are a lot of salmon in one place and they don't have the full immune system of a wild salmon, is that what you're saying? It can cause.
There's probably some stress that's involved being held, and we don't have good numbers on this. It's been really hard, but I've always felt that density is mm-hmm. very important, and an animal in captivity can't change its density, mm-hmm. right? If they're in a natural setting, they can kind of spread out, but mm-hmm. in a raceway or in a tank, they don't have too many choices, and so that probably induces some stress for them, mm-hmm. and then that's when the bacterium actually goes from being this quiescent long-term strategy, it goes into can cause acute disease, mm-hmm. which will create a lot of symptoms. And so this constantly has been a hatchery problem, and some hatcheries have it more so than others. And treating it with antibiotics has not really been it's produced some results. I know in Idaho has used antibiotics as a way of trying to reduce the level of in their spawning adults, mm-hmm. but you still, everybody has a problem with antibiotic use, including the people that do the culture. That's not their choice, but they have no other choices at that point. Hmm. So that's a case. This goes back to what I was talking about, the interaction between the host and the bacterium, right? So this bacterium, because it has a strategy where it's well evolved to live with its host throughout its lifetime, unless there's some triggering event. And actually, what's interesting is that, when salmon return, they're immunosuppressed. And that's why oftentimes you'll see salmon going upriver and they'll have big fungal patches on them. They have white patches and they look mm-hmm. terrible. And they're just investing everything in getting back to their spawning site and releasing their eggs. And so during that immunosuppression period, probably that's a trigger for Renibacterium as well to say, okay, we can start growing. But if they've successfully inserted themselves into an egg, mm-hmm. they're fine. They're just going to sit there and wait until that egg hatches. And now we have a baby fry that's already pre-infected with the bacterium.
Right. So the bacteria in its relationship with the host is about its ultimate survival, and based on whatever the external environmental conditions are for the host, it will behave in different ways. So killing the fish might be an option if it sees no other option, but mm-hmm. but otherwise it's not going to necessarily choose to do that.
It doesn't do that. Yes. In certain cases, it doesn't do that. And so that's why I talk about it being co-evolved. The mm-hmm. host tolerates it and says mm-hmm. that's fine. Actually, what this bacterium does in the host, if it's successfully established, but it's not causing clinical signs or causing disease, it actually goes inside of cells called macrophages. And mm-hmm. that's where it hides, because now instead of being out in the open and exposed to all of the immune cells that the fish has that goes around looking for bacteria that's not supposed to be there, it goes inside of one of the cells that goes around and eats up debris, and then it just hangs out inside. So it's being protected by that macrophage. And the other immune cells that conduct surveillance throughout the fish to fight off infections won't even see it, won't even recognize it. So it has a very interesting and very cool, in my opinion, very cool strategy for persistence.
Wow. Does the presence of that bacteria and other bacteria, how does that impact the, I don't know,
the long-term survival of
the long-term survival of the fish, and then also like how does that relate to like whether or not we should be eating these fish? Like, how does that?
Yeah. Don't be concerned about this bacterium infecting humans. It is one of the harder bacteria actually to grow. That's the other thing that if I'd have known at the beginning, it is a very difficult, well, not very difficult.
So for you as a scientist, it not wanting to grow is kind of hard for
problematic.
you to study? Yeah.
Yeah, right. In fact, right now, I'm actually been providing advice to a pharmaceutical company because they're trying to develop some methods for trying to keep BKD down. And the person who's doing the work is saying, having a lot of difficulty with growing it. And I run into that repeatedly. So first of all, this bacterium likes to grow at 15 degrees centigrade at a low temperature, not up around our body temperatures at all. And in fact, it can't grow above 21 degrees centigrade. So it needs the low temperature. And it doesn't have, in fact, even has different species of salmon like rainbow trout versus Chinook salmon or coho salmon or sockeye salmon. Those species have different levels of susceptibility to infection with Renibacterium. So you give a dose if you were to infect a salmon. If you were to infect a sockeye, you could use many fewer bacteria than you would if you were trying to infect, say, a steelhead. Because there's host differences in susceptibility. And if you try to infect a different species of fish altogether, wouldn't take. So that's the thing about this co-evolved. It is a very successful long-term strategy for Renibacterium, but they have a very narrow range of hosts that they can infect. And humans aren't one of them.
Yeah, just to clarify for people, they don't have to worry about that.
No.
I know that we also talked about when we met, we talked about some of the work that you were doing on kind of the I guess microbiological fingerprint for whales or health. We talked about like health blueprint.
Right.
And I was curious how microbiology can show us health blueprints for species and ecosystems, because I know that's something that maybe is like on the horizon in the field, but not fully operating at the moment. And I wanted to kind of ask you about that.
Yeah. So actually, a lot of the killer whale work sort of follows human stuff. When I was doing my graduate work, we were using what are called model organisms, right, to sort of be stand-ins for humans because we're doing biomedical research, right? And the flip side is that with killer whale, I say, the model organism is humans because there's a lot of data that's available from humans, and at least with some similarities in how we're organized physiologically and genetically, we take advantage of what's being done in humans. So my interest with the killer whales was how can we take samples, non-invasive samples from killer whale and use those samples to characterize the microbiomes that might be present, because I was aware the human microbiome project that had been going on. And I thought, okay, they're moving ahead with this, including some respiratory microbiome work. Excuse me.
No worries.
So breath is one thing. These cetaceans throw up a plume of droplets. And so the idea there was, can we look at what kind of bacteria might be present in those droplets that give us an idea about the health of the animal? So if you look at records for marine mammals in captive settings like zoos or rescue centers, like the Marine Mammal Center down in Sausalito, respiratory disease, usually respiratory infections, are the leading cause of mortality. So the idea is, well, if they've got pneumonia, and pneumonia is at the top of the list, if an animal has pneumonia, maybe there's a way for us to detect that at early stages. That was the original intent, was can we find pathogens out of these breath samples? But then, a lot of times by the time you're seeing a pathogenic bacterium in a breath sample, the animal's probably pretty far along in terms of it. But it turns out in humans, they being biomedical researchers are beginning to identify consortia of bacteria that are present in normal human breath that come up from the lungs. For a long time, they thought that everything coming out of the lungs was sterile, but it turns out that's not necessarily true. So this was just a way of pivoting from trying to identify an early sign of a pneumonia case to since we're doing virtually the same work, we're doing the DNA analysis, can we do that and identify bacteria that might be normally present in the breath samples? And are there differences with animals that seem to be not doing well? So a case in point was J50, right? The little scarlet, that little whale that she never thrived from birth. And the people who know killer whales, I don't know killer whales, but the people who do know them said that she never looked like she was doing really well. And then during her last summer here, we were able to collect breath samples from her and look at the bacterial composition. And we didn't see anything that suggested she had pneumonia or some kind of a respiratory infection, at least for bacteria. But one thing that was striking was that the complexity of the bacteria, the diversity of bacteria that were present was very, very low compared to what we'd seen with other whales. Now, she was young. She was only 4 years old. We've had samples from some other males, roughly that size, that have more diversity to it. But, just that simplicity, we're kind of wondering, is that some sort of an indication that she's not doing well? That may not have been her primary problem, right? I mean, she could have had some metabolic problem. Maybe she wasn't able to extract the most nutrients that she needed from her food. And what we're seeing is just a consequence of that other primary issue. But the idea is that, and of course, you have to have quite a few samples to start drawing conclusions about what you're seeing. And you're looking at differences in age, as well as maybe differences in reproductive status. Females of reproductive status probably look different from females that are post-reproductive, who are probably different from males. But the idea is, if we can collect these breath samples and start building catalogs of what we see in there, can they provide us with some information about the health status of the animal? Not just do they have a respiratory infection, but can they give us some indications? And that's just one tool. And there's a researcher in Canada, Sheila Thornton, who has been looking at what are called metabolites in feces. And feces is really a great source of bacteria. It's a very, very complicated community of bacteria. But what Sheila has focused on has been what kinds of byproducts, like amino acids and lipid products, does she see in the feces, which will tell you something about that whale's ability to assimilate their food. Effectively assimilate their food. So I'm just giving you that as an example that the microbiome is one approach, but the metabolomics is another approach. And so if we have people working at different levels, we at some point probably can put together what kind of bacteria are in the feces and the metabolites they see in the feces. So that was kind of my focus, was looking at mucus, which killer whales hawk up loogies and spit them out on the water, and the breath sampling, and a little bit of skin work too.
And so when we say killer whale, we're talking about orcas, right? And specifically southern resident orcas were you studying, or? That's
the focus, but we'll take breath samples from any kind of orca that is willing to share one.
Do. Okay. That's interesting. As you were talking about this, it made me wonder whether, because we talk about, in a biome sense, biodiversity is usually a sign and an indicator of health in an ecosystem and in a habitat. And is that true on a microbiological level?
As a general rule of thumb, yes, it is. But I will go back to function again, because one of the things about nature is highly redundant. So, if say a tree that produces a seed relies on a certain kind of bird to disperse those seeds around, but if that bird disappears, do the seeds not get dispersed? Well, oftentimes that tree's not relying on that one bird. It's probably relying on multiple species of birds. And so the loss of one bird may not be catastrophic for it because that function is redundantly covered by those other species of bird. And the same thing is true with the bacterial community. So biodiversity, we assume, or the diversity of bacteria, we assume is a good thing because it's a lot of legs to stand on. And if one leg gets kicked off, the whole thing doesn't just collapse. But again, not knowing completely the function of all of those bacteria that are present, it may be that, oh, this bacterium is not present in these guys, but it turns out there's another bacterium that is present that's taking that job.
Yeah. So it's still very much in the observation and discovery of function. The more you learn about the different function of bacteria, the better understanding you can have about what the redundancy is in these communities.
Right.
But I guess, I don't know if you would say this is true, but it seems like we're newer in our understanding and in our study of microorganisms than, organisms that we can see.
Yeah. Oh, absolutely. Yeah. Right.
Yeah.
Right. And that makes sense, from the human centric standpoint.
Yeah. I'm curious, what you think that people kind of, as we're rounding out this hour about microbiology, what you think it's important for people to know about microbiology just in the everyday, and what we can learn from viewing the world from a micro perspective.
My first message is bacteria are not bad. Okay. I mean, they are your friends and you need them. And if you're getting an infection, that probably says more about your own inherent health status than it does about your exposure to something, right? Because these guys are probably always around. So I'm for being clean. I agree that having clean water to drink and washing your hands, all of that stuff is important stuff, but don't feel like you have to be sterile. In fact, being sterile is not really what you want. And I think there's been increasing evidence that maybe some autoimmune diseases might be a result of not getting the kind of diverse exposure to microbes early in life. That's a speculative link, but there's data building up to that. So they're your friends, and don't feel like you need to sterilize them or get rid of all of them. But you do need to be careful. I mean, they can cause problems. And then what was your other question?
What can we learn, just philosophically speaking? What do you think the value is of looking at the world from a micro perspective?
I don't know for other people what that might be, but for me personally, bacteria and microbes were here billions of years before us. They will be here long after we are gone. It gives me that long view that I know that they're there and that they will do well. They will adapt to whatever situation we create or ends up being created. So I think it gives me some comfort because I know they're going to be around and they'll continue to represent biology on the planet until we collide with the sun or whatever's supposed to happen.
Yeah, they're ancient life forms. I think that's
they're ancient and future life forms. Very very adaptable. I think if you wanted to convey some sort of philosophical bent to other people, they are incredibly adaptable. Not all of them survive. They do play. It's a numbers game. Definitely a numbers game for them, but they do respond to environmental change and they adapt. And I think if you wanted to have a mascot for adaptation, you could be looking at microbes.
Wow.
Or particularly bacteria.
Yeah. Wow. Well, thank you so much. That was really enlightening. And I really appreciate you sharing all of that knowledge with us.
Okay. Hopefully it wasn't too detailed, but
No,
fun
I
to
think it
share.
was great. I think it was great. And if people have any questions or if they want to learn more about DNA and RNA and, open up a biology textbook and get it out or go to Google and
Absolutely.
hopefully it'll spur some curiosity. That's my hope.
Absolutely.
And I love that.
Another man up in the pulpit in the state house. Head of the table with a black pen, scratching egos, talking too loud. Externalize every outcome, every warm gun, every profit. And kneeling down to the flashing numbers, the NASDAQ, the cold constructs. Running backwards to just keep moving, just keep moving, just keep moving. Unlearning every lesson that our mothers tried to teach us.
This podcast is a production of the Whidbey Environmental Action Network and is made possible by our generous donors. Produced, edited, and mixed by me, Amanda Bullis. Special thanks to Marianne and Steve for their years of service through action and for lending their voices to this podcast. To WEEN's Executive Director, Marnie Jackson, and our board for their leadership. Special thanks to Whidbey musician and WEEN board member, Nathaniel Talbot, for letting us use his track, Intuition, from the album Animal, available on Bandcamp.
Every day on your paycheck, a volume of confidence against the idea of your body being put into your own hands. So please don't hold it, please don't hold it, please don't hold it back. Unleash the power, the straight logic of compassion. What I need now ain't that old religion or the cold hard facts. Just need a woman's intuition, get us on track. I just need a woman's intuition, get us on track. Stepping out of the shadow of a false man's allegory, slowly erasing that old violence from the story. Recoupling emotion, slow emotion to intelligence, granting the planters, healers, teachers, feelers a new relevance. What I need now ain't that old religion or the cold